He kōna I purangi tēnei nā te reo irirangi o Aotearoa.
Kia ora, nau mai haramai ki te ao hurihanga. Hello and welcome to Our Changing World. Ko Claire Cannon tēnei. This week, we joined some botanists during their first ever attempt to reintroduce a threatened native orchid back into the wild. Now, New Zealand's native orchids are nothing like the colourful, showy plants you find in the shops. They're generally less in your face. And this one, a potato orchid known as Cooper's orchid, takes this to the extreme. It lives mostly underground and only emerges briefly every few years to push out a kind of drab-looking flower. Like all orchids, Cooper's orchid only survives with a lot of help from a fungus, and that's what makes orchid conservation such a challenging job. But this is one of our most elusive and rarest native species. Veronica Meduna has this story, and first she catches up with botany curator Dr. Carlos Lenebach for a botanical orchid tour at Te Papa Tongarewa.
It's a massive group of plants, the second largest group of flowering plants in the world. They're beautiful flowers, but when you look at their seeds, they're very simple. The seeds are just basically a naked embryo. There's no food stored inside the seed. So the only way that the development of that embryo will be triggered is when the right type of fungus is on the soil and that infects the embryo. There are about 25 to 30,000 species in the world, and they're found basically in every single continent except Antarctica and in deserts, but they're everywhere. When you think about orchids, you think about big flowers, colorful, very showy and attractive looking plants and flowers. But when you go to places like here in New Zealand, and especially southern New Zealand, you'll see some of our orchids are very small flowers. The colors are brown, green, purple, dark purple.
It's this unassuming nature of New Zealand orchids that convinced Dr. Carlos Lenebach to leave his native Chile and move here. In his role as the botany curator at the National Museum, Te Papa Tongarewa, he studies the peculiar ecology of orchids and has described new endemic species. But recently, his focus has been on one particularly odd group known as potato orchids.
And here we are in the botany collection area of Te Papa, and in here we have about 300,000 plus specimens of plants that have been pressed.
Te Papa's herbarium is like a huge warehouse. Movable shelves rise from the floor almost to the ceiling, all stacked with boxes and files of dried plants. As we walk past the preserved remains of subantarctic mega-herbs, we reach the potato orchid files.
This is a very unique group of orchids that we have here in New Zealand and in other parts as well of Asia. And also in Australia, this genus is called Gastrodia, so the common name is the potato orchid. There are about 50 species of Gastrodia across Asia, the Pacific, Australia and New Zealand. And they basically look like, at least the New Zealand ones, like a stick, like a dry stick that is poking up the leaf litter, and you would not see them unless they're flowering. No leaves, there's no green in them, they cannot produce their own food, and they just rely completely on fungus. So these are plants that basically never leave their baby behavior. They never reach a stage where they're completely independent of their fungus.
The dried specimens really are no more than a stick with small brown flowers. There are 5 species of Gastrodia in New Zealand, and they are known as potato orchids because except for that brief period when they push out this flower stick, they spend their life underground, growing in small tubers much like kumara or dahlias.
So we've got here a folder. We have several specimens, and when you look in there, that structure in there is what we call the tuber. So these tubers are about the thickness of my thumb, for example. There's another species, Gastrodia sesamoides, that produce bigger tubers. What's quite interesting with this is people in Asia list some of the most abundant species, Gastrodia elata, is actually digged out and is cultivated, and people using it for a number of purposes. So it's medicinal, but also they're using it for ice cream or for put it in your chicken broth and for tea. There's a big market for the Gastrodia elata in Asia. In New Zealand, the tubers were consumed in the past by Maori. They would roast it and eat it. I've never tasted one. I don't know if I want to go there. To have a good meal, you would have to collect a lot of this plant. But this plant is quite abundant, so I would not see that as a big effect on the population, natural populations.
It's a different story, though, for one particular Gastrodia orchid. Almost 10 years ago, Carlos and his colleagues identified and named a new species in this group, Gastrodia cooperi, or Cooper's orchid, named after botanist and orchid lover Dorothy Cooper. At the time, there were only 2 known populations of this potato orchid, one in Nelson and one in the Wairarapa, where only 19 plants were found. Even after more patches with Cooper's orchids were discovered, there were fewer than 250 plants across New Zealand, and it was declared nationally critical, the highest threat classification. But how do you try to save a rare orchid given they need a fungus to even start producing a seedling? That's when in vitro fertilization comes in. Let me take you to a small lab at New Zealand's only native botanical gardens, Otari Wilton's Bush in Wellington.
Good morning, my name is Karen van der Walt and I am here at the Lions Otari Plant Conservation Laboratory. So please come in.
Oh wow, this is quite a nice room. So
Yes, so this is a laboratory, or it's an old flat that we've converted into a laboratory, and it's got all the cool equipment, science equipment that we can use to study and understand and conserve our native plant species. And working with me in the laboratory is Jennifer Alderton Moss, and she's going to show you some of the potato orchids that we have been propagating for the past 3 to 4 years.
Excellent,
Jennifer.
let's have a look.
Our potato orchids are kept at the moment in this really high-tech warehouse cabinet.
Is it temperature controlled?
It's not temperature controlled, it's really just a metal cabinet so that they stay in the dark, because that's the only thing that they're particularly fussy about, but the lab itself is temperature controlled, so it's always 18 to 20 degrees in here, which is really nice for me and for the orchids.
So the room is temperature controlled. You've got some incubators, but for the
Gastrodia, we just keep them at room temperature and they seem happy enough.
and the darkness is because they live underground usually. So
I mean, there's no need for them to have light. The tubers live underground. They only come up to flower, and when they flower, they have no leaves and they have no chlorophyll, so they don't need the light for anything.
So let's have a look.
Yeah, so we've got in this plastic bag.
And so they're all in petri dishes.
They're all stacked in petri dishes. The way that we have them grown at the moment is they're on agar, so just a gelled media, and then we feed the fungus that's with them these leaves.
They look like beech leaves.
They are beech leaves, so we're feeding our fungus beech leaves, and the fungus will break those down. You can sort of see that these ones are starting to go pale and get bleached by the fungus.
So I can't see the fungus itself is obviously microscopic. So what I'm seeing is the leaves being
munched away. So it's getting these white spots where the fungus is eating the leaf, and then it uses the leaf to feed the plants.
Yeah, and where's the orchid in this?
So the orchids are these little yellowy white blobs, for lack of a better word, blobs, sticks, little noodles, what do we call them?
Oh, noodle is a good description.
Yeah. Sort of.
So whitish, opaque.
Yeah, little pale,
noodly
branchy. Yeah.
Tiny.
So they're just small tubers at this stage.
So can you talk me through the process? Because this is essentially, it's like an in vitro production of the seedlings, which would normally happen in the wild, but you start with literally just the seed. You throw the seeds on these plates?
Yeah, so we start with literally just seeds. We're actually growing the fungus out first so that the seed can go onto where it's already got the fungus. The seed's about 0.5 millimetre long, So very small, and the embryo is probably about 200 micrometers. That's really what needs the fungus. So it's a really tiny embryo. It doesn't have the nutrients that it needs for itself, so it needs to form that association with the fungus. And with these ones, they just keep that association their whole lives.
Is there something we could possibly look at under the microscope?
Yeah. So we use the microscope for when they're still really small, because obviously the seeds start off tiny. And the microscope we use just to check if they are growing, in what conditions they're growing, and how quickly and things like that. So I'm going to show you some of our baby Gastrodia cooperi. So if you want to have a look there, you should be able to see the white threads with the sort of crystal structures on them. They're called astrocystidia. But that is the fungus. So the thin white threads are the fungus.
So that's like as you imagine, the sort of hyphae of a fungus, the threads that a fungus produces, okay?
And the yellowy bits are the seed. So it's an embryo in like quite a long, skinny seed case.
So they're literally egg-shaped things that I'm seeing?
And some of them you can see have started to swell.
Yeah. Mm-hmm.
And
so that's the ones that are starting to take in the fungus?
Those ones that are bigger than the normal seeds are ones that are taking in the fungus and are starting to germinate.
There's one that's quite swollen and has a lot of those fungal threads around it. So do they physically have to move towards the seed, the fungus?
Yeah. Orchid mycorrhiza is an endomycorrhiza, which means it goes inside the cells of the plant. So the fungus will grow over the seed and into the cells of the seed, and then that interface will be where the seed gets nutrients from it and allows it to grow.
So that's what we're seeing in the middle of this image here. There's a big
sort of a net of fungus over the seed almost.
Yeah, and the biggest seed is in the middle of that, and it's all wrapped up with.
Yeah.
I was going to say it looks like a piece of food in my fridge when it's all full of fungus, but
it is very much the same thing as what's happening with a piece of food in your fridge, is that the fungus has found somewhere that it likes and it's growing over there, and the orchids are maybe taking a little bit of advantage of that.
Excellent. This is quite amazing to see them at that very first stage of their growth. Really.
I think they're really beautiful. I think the fungus is really beautiful too, actually.
The team came up with the idea of using in vitro processes back in 2019. But first they had to search for the plants, monitor them to figure out the best time to collect seeds, identify the right fungus, and get the funding for the lab work.
And we germinated them. The first time was it in 2023 that we got the protocols going. And then from there, it's obviously, you can see the progress going on. But leading up to it, it's been a lot of intensive hard work and not much success.
Well, up to now, though.
Up to now, exactly, until we found the right fungus. And that was also a couple of attempts until we got the right one and then got going from there.
And so now you're ready to return them?
That's right, yes, because like you just saw in the microscope, they are still small, so even from there we have to figure out how do we get them bigger. Do we continue feeding them with beech leaves? Do they continue relying on that fungus we give them, or are they going to look for a different fungus, which is sort of what studies from China indicate, that they will switch partners later on in life. They won't be able to sustain that. Fortunately for us, they have actually liked this fungus we've given them and have grown into fairly big tubers that we are now ready to take back to the site that we went to in December 2021 to find these plants and the capsules.
The big day came in late January. Equipped with shovels and picks and a small plastic box stacked with orchid seedlings, the team returned to the site where they'd first collected the seeds. This was the first attempt to return a threatened species to the wild. Here's Carlos Lenebach again.
We just walked up about 10-15 minutes up a hill, and we are about 20 kilometers away from Masterton in the Wairarapa. And this is a quite interesting spot for New Zealand flora because this is the type locality of Gastrodia cooperi. So the type locality means the specimen that I used in collaboration with Jeremy Rolfe to describe this plant as a new species was collected. So that specimen came from this area. Today is quite special because it's the very first time that we attempt to reintroduce seedlings of this potato orchid, a Cooper's orchid, into its natural habitat. Many years ago, when we first described this species in 2016 as a new species, we noticed that the population was very small, and that the place where this population is found is surrounded by farmland. So habitat protection and habitat transformation in this area is quite a thing to be worried about. Luckily, there's a nice piece of bush in here that is protected, and that allows us to be able to come here every year, look at these orchids, collect their seeds, and study the biology of the plant. Unlike many other plants out there in the world that are threatened, orchids are very fussy because they depend on a number of interactions with other organisms. So we have, the most important is with the mycorrhizal fungi. The other thing, they have sometimes very specific associations with pollinators. So knowing what insect or what other organism is in charge of the pollination of the flowers is very important, because then you can make sure that that organism is also present at the site and that you get good number of fruits produced per year, and therefore good number of seeds and good quality of seeds that you can work with and propagate.
As we clamber over fences and down slippery paths, it's my turn to spot a brown orchid stick among a sea of other brown sticks. I'm hoping that it's one of those things that once you've seen one, you start actually recognizing them or seeing them, right? Oh, so this is no.
This is a brown stick.
I'm still easily fooled, right? That's not it.
The orchid. I generally refer to it as a stick, a brown stick in the forest floor. These sticks, though, are about a meter or more high, and they will have like 20 or 30 flowers on the stem.
Still fresh flowers or wilting flowers?
At this stage, we're hoping to find capsules with seeds in it. In them, and we're hoping to use those seeds, get some of those seeds and take them to Ottari Wilton's bush for germination experiments.
Cool. Are you saying you're seeing one there where that... Oh my God, okay. Okay, that would be so easy to not even spot.
Yes.
Oh, it's a really sort of just brown, dark greenish flowers. It does look like an orchid flower, so once you actually look more closely, you would think orchid, right?
Yeah, it has that sort of like basic structure of an orchid. So it's got 3 sepals and 3 petals, and one of those petals, it's very highly modified, and it's internal in this case. So when they're just starting to come out of the ground floor, you look at them and they remind me of like a little asparagus coming up from among the leaf litter. And you can see they're brown, there's no leaves at all. And this plant, which is just finishing flowering, has got about 10 flowers. And that lower part down there, that looks a little bit thick, that's the ovary. So if we want to collect seeds in this case, we will have to come back probably mid-February, because these are not ready. But if the flowers have been fertilized, that structure, the ovary, will look very thick and plump and fat basically, and we have to be here at the right time to collect the seeds before the capsules open. The seeds otherwise will just be dispersed by wind and gone who knows where.
While the rest of the team stakes out the best spots to put the seedlings into the ground, Karen checks the site for flowering orchids to collect more seeds.
So we only need to collect 1 or 2 capsules in order to get a lot of seed to work with, which is great for the orchids. So we always leave, by far, we'd never take more than 10% of the fruits from the population, so the seeds go back in any way. And that's what I'm doing now. So I'm jotting down how many flowers, how many capsules do we see on the specific plant, and then when we come back in 2 or 3 weeks time, we can get an idea of what works in terms of the plant. So if we see 5 flowers and no fruits, we know this plant did produce flowers but it didn't make the capsules. And then as you can see, a lot of the labels have got no plants with them. That is because we don't understand the ecology of these plants or the biology, but it doesn't seem like they come up every year, so they skip. Some of these we haven't seen for 3 years, so we've seen them once and not again. So it just gives us an idea of what's going on. How many plants are we looking at in this population so that we can effectively work on them?
The team managed to produce more than 60 tubers in the lab, but they're so precious. Only a few are being planted today.
We are returning 14 of those today. And even that is a process of learning. So how exactly are we going to do this? Where are we going to plant them? Are they going to survive? How are we going to figure out if they survived? So because these plants don't pop up every year, how are we going to know?
How to monitor.
How to monitor is not very straightforward.
The seasons might be important for them, but it's obviously not the only factor that triggers
We don't
fruiting
have that
or
answer,
flowering.
but we have had reports. So we've been working on this orchid. We've been talking to Doc and other orchid people on South Island as well. And we've had really good feedback this year from this potato orchid that it seems to be a good year for them. Whatever made it a good year, I don't know, but there's a lot of reports of them being observed in different areas. So obviously the climate leading up to this time was good for them. Yeah. But we don't have, and that's the challenge for us when we work with these really rare species. There's such a massive knowledge gap that it becomes really hard to try and it's almost like the ambulance is reaching the bottom of the cliff already. And now we've got this whole mountain of information to try and figure out before we can effectively conserve these species or prevent extinction.
Shall we put one down?
Yeah.
We move carefully. Only a few orchids are in flower, but we don't want to disturb the tubers growing in the ground below our feet.
Careful here to not tread on them. Eh?
And then it's finally Jennifer's turn to let the seedlings go.
You carried in a box of precious goods, precious cargo.
I've got the little chili bin, and in here we have some culture vessels with orchids in them.
So what's the stuff on top? That just looks like wood shavings?
It
or
is.
some sort?
It's just wood shavings. And so the wood shaving layer or the leaf layer that we have in some of them, it's just a carbon source for the fungus. So the fungus will eat the wood or the leaves, and then it will transfer that to the.
Have you got some with a leaf collected from here, from the side?
No. So we've been mostly using. Sorry, ignore all the jingling. Beech leaves. We find that. We originally started them because they're pretty regular in size, so we can control how much we're feeding the fungus at each time, but it seems to like them more than the pine shavings that we give them anyway.
So what we're seeing here is the little tubers.
Yeah,
they are
so
still
we've got
quite
some
thin and short,
The
right?
little baby tubers, I guess. So they're sort of at the stage, they'd be 2 millimeters width, and these little branching forms.
Yeah.
Sort of wormy, could be like little hands or something. They're very alien looking.
So, for the orchid, it's quite clear what's in it for it. It's the food supply, but what's in it for the fungus, do you know?
It's something that's not been very well explored. In green orchids, there's been a couple of studies that show that there is some, for the adult plants, they do transfer some nutrients back, but it's definitely an under-researched field. I don't have a very good answer for you about
lots of research
why a
to come.
fungus would go into a plant that is never going to give it anything, because obviously these don't photosynthesize. There won't ever be a stage in their life where they'll be transferring nutrients the other way.
But in your work, you were able to isolate the fungus and grow it, so it does not need the orchid to survive itself.
Orchid,
right?
so it can live off decomposing material. So it'll just live in a wood pile or something, for example.
So now the decision will be where to put these. They've grown in the lab, they've grown well enough with this fungus.
I'm pretty reluctant to put them out myself because they're my babies, but...
Do you have that sense of protection of them? You don't want to let them go?
Definitely.
And you won't know
we
how
know
well
that
they do,
grow
right?
here as adults, right? So this is going to be the best place for them, but it's a little bit sad.
And you won't know how well they do for quite a while because they may not flower for years.
We
right?
have no idea how long it'll take for them to flower, I think. I'm not sure if you've read the book The Orchid Thief, but he talks about I think it's a ghost orchid, it's one in North America, but they say that it takes 7 years before it flowers. And that's one that photosynthesizes. So we just have no idea, no clue how long it'll take.
Like so many living things here in Aotearoa, these orchids are found nowhere else in the world. And while you're unlikely to see them in a pot on your office mate's desk or in flower shops, they have their own brown stick-like charm, playing their part in the native ecosystem. When everything's done, all that's left to do is wait.
We're really hoping that it's going to work. We don't know. There's a lot of unknowns with working with orchid conservation. Lots of questions, but we're very happy that we've managed to figure out a system where we can grow the seeds and have seedlings and then being able to reintroduce them to this site and perhaps to other sites as well. So if something bad happens to this, like a big catastrophic event that wipes out the population, there's a landslip, whatever, we will have a backup that can help us to reintroduce the species back to the wild.
Thanks to Veronica Meduna, who reported and produced this episode. Veronica spoke to Dr. Karen van der Walt and Jennifer Alderton-Moss at Otari Wilson's Bush, and to Dr. Carlos Lenebach at Te Papa Tongarewa. Production help was by Ellen Rikers and sound engineering was by William Saunders. You can learn more about this topic on our webpage at rnz.co.nz/OCW. Tēnā koe I whakarongo mai. Thank you so much for listening. I'm Claire Kincannon. Have a great week. Kia pai, te wiki.